Okay, I was between two titles for this talk. The first one was "Don't say I didn't tell you so," or "Don't say I didn't warn you about doing automated science." But with this one, I'll explain what the title means in a bit. So we've been doing closed-loop automated science at Michigan for a number of years now. We've done about 1.5 million closed-loop experiments. Most of those have been designed by AI and carried out by robots. And we started to have this great partnership with people at PNNL and exchanging lots of ideas both ways. So for this talk, I'm going to spend less time focusing on the science that we've done. I'll show some examples of things, but really stepping back and saying, what have we learned doing all these experiments, spending all this time trying to do science with automation without humans, and putting that in a framework that we think is very helpful for structuring these types of projects. So I hope this is useful. I thought it would be a good presentation for a workshop rather than for a scientific conference. The title is actually in reference to this thing called Moravec's paradox. So this was coined in 1988. And what they said is when you're thinking about AI, that things that require humans to stop and really think about something turn out to be very easy for AI systems to do. But things that we do instantaneously, almost without thinking, are really difficult for AI systems to do. So here's a couple examples. A few weeks ago, I drove down to Indianapolis for a conference, and I have to do two things. So one, I need to find the route to get to Indianapolis from where I live outside of Ann Arbor. And this I put into a mapping system. This is a type of AI thing that planned the route and said, here's the best route. You could even fly if you wanted to from Ann Arbor to Indianapolis. It's just a 4-hour drive. So for me to plan the route would take me to sit down and have to find an atlas. I'd have to look at the maps. I'd have to make a bunch of decisions. It would be a long, slow process. But this problem has been solved by computers since the late 90s when we had MapQuest. I found out my father still uses MapQuest. I think they keep the site up for him. He showed up with printed directions at MapQuest at my house one time when he drove in. Right? So that's been solved by AI for almost coming up on 30 years now. After I had my route, I got in my car and I drove to Indianapolis. And driving is thousands and thousands of instantaneous reactions that you don't even think about. There were some people I drove by in Indiana who were not thinking about their driving at all. So driving cars, although we can get any 16-year-old to learn how to drive a car or to ride bicycles, is still not something that AIs have mastered. Even though it's not something that requires lots of deep thought, it requires this instantaneous thought. That's Moravec's paradox. Another one is that Amazon is really good at shipping things to you. So when you buy something, they will preemptively ship other items to the fulfillment center, meaning when things come into Amazon's supply chain system, they will put it at a storage facility based on where people have buy products that mean they're going to buy other products. So if you buy something that has a consumable and they get a new consumable in, they will put it by where lots of people are buying it. That's been handled, it's been going on for 10 or 15 years. They've had this AI that can predict what you want to buy. It would be very difficult when I'm trying to buy someone a gift. I have to stop and think, what should I get this person? Amazon solved that. But inside Amazon's warehouses, the one thing they still have people do is pick stuff up, right? Because this is a very difficult task to figure out. When I pick up this Kleenex box, I use very, very different dexterity and different amounts of pressure than when I pick up this marker if I were trying to pick up a chair, especially because they're bolted down in this room. So that is instantaneous and is something we still haven't figured out. In fact, there's a little company called OpenAI that you might have heard of. They, in 2021, shut down their robotics unit. They had a physical robotics team. They had robot arms that would do the grasp task. They would solve Rubik's Cubes. They had a room full of robot arms trying to pick up stuff. And they canned the whole project, saying that this is a little too difficult. And, quote, they've shifted their focus to other domains where data is more readily available, i.e., language models. So obviously some talented AI people at OpenAI, but they said the thing we can all do instantaneously doesn't show up. Right? So I'm really glad that PNNL has decided to focus on the things that humans do instantaneously. Right? We can pipette, we can move plates. I worked in a lab for 10 years. I had one pick and place error. I remember I dropped a plate one time and it stuck with me. Like one plate in 10 years is pretty good, but for robots, that's quite a challenge. So PNNL is now venturing in to fight Moravec's paradox and say, we're going to solve these tasks that are easy for humans, but we're going to do it with robots. And so when we think about automation, we first take the standard scientific process and then figure out where the automation comes in. And the Society for Automotive Engineers, they have 5 levels of autonomy for driving, all the way from you're driving by yourself, you use cruise control, adaptive cruise control, lane keeping. In the future, there will be a fully self-driving car. They have these all mapped out, and we thought we should map out the same thing for automated science. But first, let's go through how science works with humans. So we start with some sort of prior knowledge. We've read a bunch of papers, and we decide it would be interesting if we studied this microbe, right? That formulation to say we are going to study this pathway in this microbe by analyzing it in these conditions, we have formed the problem now. We've narrowed it down and said this is the strain, this is about what we're going to study. For instance, we're going to study raffinose, and we're going to interrogate this gene, and we're going to use growth assays, right? After the problem is defined, then we actually set up the experiment. We said we're going to grow these strains, we're going to make these knockouts, we're going to grow in this amount of raffinose, and we're going to measure this as the output, the growth curve. Then it gets sent to 3 things that actually happen in the lab. This all happens at your desk, right? Some people have their desks in the lab, so it feels like they're getting more lab work done, but most of this time could be done in the library. So first it goes to scheduling, where you either program a robot to actually carry out the experiments that you've dreamed up, or you plan out in your lab notebook. I have a picture of the 96-well plate, I'm labeling all my tubes, this is the scheduling problem. Then it happens, you either have a robot do the pipetting or you do the pipetting, and then finally there's some sort of data processing that happens, where it goes from raw data into things you can actually interpret. When all that finishes, you're left with some sort of new data. Now I have growth curves, they've been normalized, I've looked at the controls, it's passed the QC filter, and from these experiments, I can create new knowledge, and I can update my belief about how this system is wired. Okay, so that is how we view the scientific process that happens along these steps. Any questions before we go? All right. Interrupt me if you have any questions that we're going through. Now, the key to this whole process is that it runs in a loop. So the new knowledge that comes in becomes prior knowledge, and then you can run through the cycle again. Right? To get a PhD, it takes about 200 experiments. The tricky part is we don't know what those experiments are, and that's why it takes 5 years. And we don't do a year of experiments and then 4 years of analyzing the data. It has to run in a loop. So autonomy is about getting back to this loop without humans actually moving through the loop. Okay, I'm a little scared because this line seems to have cut off the bottom, so I'm worried that all the rest of the animations are going to be a bit off, so you have to forgive me on that. Okay, but here's the first one. So level 0 autonomy, also called lab automation, is we bought a robot. We bought a robot, and the problem is that covers one step of this, right? We now have programmed the robot, and the robot does things in the lab, and then we have the raw data that comes off the robot. That's the first level of autonomy that we get, right? The sort of second level of autonomy is if you have an experiment design and we can translate that into something that goes on a robot, and then we can handle the QC on the data. Now, neither of these two solve the closed loop problem, right? Robots sit there until humans tell them what to do. So getting a robot will only speed up your experiment if the bottleneck in your process was going in and physically pipetting in the lab. If you have data you haven't analyzed or experiments you haven't planned, these are not going to make your life any faster, right? Because they don't form a loop. So the loop can't run any faster, right? Where it actually can form a loop is when we get to level 2 autonomy, where we take the new data and now have an AI that says, well, here's the next experiment to do. All right, that requires some sort of model. And this level 2 is where our lab spends a lot of our time, this autonomy. Bayesian optimization is this level 2 autonomy. Right, now we can speed it up because this short circuit here runs without any humans down here. We can just run the cycle and let it turn the crank of new experiments and keep the robots moving even without humans stepping in. The last part that probably only became possible recently is if you can take prior knowledge and then say this is a good thing to study. And it wasn't until we had the language models where people started to think that this is a possibility. Tell me what would be a good experiment to do. Could you write a grant application for the NIH that says that that's level 3 autonomy? This is where the real dystopian stuff comes in right here, okay? So I'm going to step through these levels and tell you about the things that we've learned going through each one of these levels, some of our big lessons. Okay. The first one is when you talk about level 0 autonomy, when you bring robots into the lab or buy a whole bunch of robots, things change and the work that scientists do really changes. And this is more of a social issue than a technical issue. There's this fantastic paper by a group led by a researcher out of France, where they studied all the biofoundries that were spun up in the United Kingdom. And they didn't look at productivity. It wasn't a quantitative study. They interviewed the people working in there and say, how did your life change once robots came into the lab? And the results were quite striking. One thing they said is when junior researchers realize they have to do so much optimization, meaning to get protocols to run, if they want it to go on the robots, they usually give up. They think, let's just use it like it's an iPad and touchscreen, but it's not quite like that. You still have to do all the prep work. You have to do all the preparation. You need to set everything up. Another participant said, the time taken to set up automated processes is often not worth the time saved in carrying out routine tasks. The less complex systems are easy to set up, but they're not worth the time in automation. Right? One of the big take-home points of this is that everyone said, when I came into the wet lab as a scientist, for the first week, I learned how to pipette. I had someone tell me, you need to empty the tip before you eject it. You need to, all these little tricks. And then I never thought about pipetting ever again for the rest of my scientific career. I could stand and talk to someone else in the lab while I'm pipetting, while I'm opening tubes, while I'm moving plates around. There was no thought in it whatsoever. As soon as automation comes in, I spent my PhD staring at a robot moving liquids, like babysitting and watching it, and all I thought about is pipetting. Right? The work goes from the high-level work of I'm a scientist, I'm designing experiments, to I'm a babysitter for lab robots. I was going to have a joke about the movie, the men who stare at goats, and it was going to be the scientists who stare at robots. I thought that's a joke that either Scott will like or Justin will like, but not both of them somehow. And then Ben told me that this was a bad idea, so I pulled the joke in the end. I thought, now I'm just going to describe it in a half-hearted way, and then no one will laugh at the joke. That'll be much better. But there's a huge, like when you bring in robots to the lab, what it means to be a scientist changes, and you automatically separate between you have high-level tasks and just planning, and then everyone else is focused on these mundane things that they did not think about when they signed up to be a scientist. I didn't sign up to be a scientist to worry, did it really aspirate? Did it not? Why did it put, it can move the plate there. It has room. The robot says it doesn't, but it doesn't. Okay? The second thing was already brought up again. So talking about quality control. When you're doing this level 0 autonomy, you want to put things off to the robots. Humans naturally know when they mispipetted. They say, wait a second, that doesn't look like 10 microliters. No, the robots don't have this capability yet. And you need to QC the instruments all the time. This is actually a Formulatrix Tempest. This is the same instrument that's going into the Ginkgo one. It uses these microfluidic chips in order to fire droplets into the cell. So there's each one of these tubes is hooked up to a chip right in there. We have 2 of these Tempests in the lab, and we have to QC these twice a day. So we have QC assays that go in to check every nozzle of the Tempest because they get clogged. And these are controlled charts that get run. And you see this is one of our chips on one of the days got clogged, and we can tell with this spike in the optical density when we read out on the plate. And we know that we have to throw out the data from this, but we do the QC before we run the instrument, and then we do the QC after. And if it fails the after, then we have to cancel all of those experiments. But there's 96 nozzles on this thing. The dispense volumes are 0.1 or 0.5 microliters. So you can't tell when it misses dispense. We have to have these procedures baked in for all the instruments. They have to run every day. And the process of looking at these has to be automated. Otherwise, you have a full-time technician just staring at hundreds and hundreds of QC things for all the different instruments. So like they said in the UK, it might not be worth the effort in order to do the automation. Okay. But so now we can step up to level 1. So level 0 is we got a robot, and now we thought things were going to faster. Level 1 is to say, I spent all my time programming a robot to do something I could just go pick up a pipette and do, and to do QC on the data that comes off the robot. So if you automate that, that's level 1 autonomy. Now level 1 flows through level 0. So we have some sort of AI system that schedules and plans the robot, and then something that takes the data and does the raw normalization offer, but it uses a robot in the middle, or it tells tells a human what to do in the middle. So this is level 1 autonomy, right? Here is our experimental setup. So we get some sort of experimental design. We have different runs. We have different concentrations of reagents. They go to a scheduling software. This is not an AI system. This is a deterministic optimization and scheduling procedure. It gets sent to one set of robots that set up reagents and plates just to go to other robots that do the combinatorial dispensing. And then we go to the bulk filling. If you have common media to all of them, then it goes through some robotic incubators that pass things in and out of plate readers again. So we focus mostly on microbial growth. When we started, we could do about 300 experiments a day. This summer, we're doing some upgrades to the system, so we'll be able to start 60,000 experiments every day. So our throughput will be a little higher than the big M2 PC system, but we are much less flexible. So we are focused on growth. We can do lots of growth experiments because the system is designed primarily for growth, but we don't have, let's say, the synthetic biology capabilities that the big system does. So there's a flexibility, efficiency trade-off in between the two systems. So when the user says, I want to do these experiments, I want this concentration of this thing in the well, we have to schedule and translate across all of these robots and the plate movements going back and forth between everything. And this is the level 1 problem. You have an experimental design that comes in from the user, and you want data that comes back to the user. How do you do that scheduling? Ben David, who's right there, Ben can raise his hand, is going to defend his PhD. Coming up, he is the expert in our level 1 automation. So any questions on how this happens, you can talk to Ben, and Ben will be glad to correct all the things I'm going to missay in the next slide about how this actually works. There's a duality between the world that the scientists live in and the automation engineers. So when you talk to the automation engineers, or you look at the really nice protocol building software that Ginkgo just showed, right? They think about things in automation-centric manner. Pick up a tip, go to well A3, aspirate this volume, and put it in well A9, right, of this different plate, or move the plate. And the scientists say, like, well, I don't care where it was. I don't care what plate it was, if it was in a tube or if it was in a Pyrex mixing bowl. Like, all I care about is that in the end, I want an experiment where you grew this bug in this media with this concentration. And oh, by the way, in my lab, I have stocks that are already made with this concentration. So that's what you can use. So you can think about scheduling in this sense. How do I mix these stocks that I have into this thing that I want? And I don't care how you do it. It just has to happen. Or you can think about, I have these wells and I really don't know what's in them, but I know that I'm going to use this robot at this time with this tip to move it into a different well. Right? This is the engineering view and this is the science view. And unfortunately, a lot of the friction in automation is you have the two people, the scientists are coming up with things that are not easy to do on the engineering side or vice versa. So Ben's thesis was actually to say, how do you map between these two sides? Because we're not going to get the scientists to think like engineers, and we have to not have engineers involved in every scientific decision. So he came up with a piece of software called the scheduler that basically maps between the two sides. So given what we have in the lab, it knows all of the reagents that we have, all the bacteria that we have, and given sort of the new state that we want, what do you want the final experiments to look like? What do you want in those wells? What bacteria do you want to grow? And it then can output what are the physical operations, the protocols, the schedules that happen. Well, you should use this robot and you need to put it in this plate and you need to move this plate between the two, right? And it's not a one-way flow between these. It actually connects all three of them with a single mathematical framework. So one way you can run it is to say, if the scientists told me they want these assays to happen, and they're going to happen in some plate, so I then can say, I'm going to use these robots. These are the 16 robots we have in the lab. These are what you can use. The scheduler can say, if you're going to do that, then you need starting reagents like this. For instance, it says, well, you have a 50 mil conical of this antibiotic solution, but the robot you're going to use can't draw out a 50 mil conical, so you have to move it into a deep well plate ahead of time. It can solve those types of problems. Okay? Another way you can run it is to say, if you know what's in the lab and you have results that you want, you have a set of experiments that you want to do, it will then write you the protocols for each robot in the sequence that it needs to happen. It says load this plate, put on these tips, and it can translate that into liquid transfers that actually happen. This is the most common way that the scheduler is run. It says, given what we have in the lab, here's what you need to do. The last way I think is the most interesting because best laid plans never really happen. So you get all these protocols and there's some sort of error. You find out afterwards that, wait a second, this wasn't cysteine, this is cysteine. And I don't know who named those things to be so similar to one another and put them both in the defined media for streptococci. I would like to meet that individual and we will have a spirited discussion on that. But it worked. You say like, well, will we put cysteine on the robot instead of cysteine? All right, so now we have all of this grand scheduling to say, here's the results, here's what's in all these wells, but the technician came back and said, I mixed something up. Right? So we can actually recalculate the state by saying, I know I was supposed to do these physical operations, but one of those was to load cysteine on the robot, and I loaded cysteine instead. So given what I started with and now what actually happened in the lab, according to the process log, it will say, well, what's actually in your experiments is this. Who cares what you thought you were going to do? You made these reagents. You can love it or list it type thing. That's what's in the well. So we can update our planning after we've done it. Okay. So it turns out all of these uses one optimization software, and it's whatever variables you fix, you solve for the other variables. So it's the same problem. Pick any two, fix them, and you can calculate what the third is going to be. And Ben will be happy to talk to you about all the work that he's done to developing the scheduling software. Okay? The other thing to be wary of is that there's this anti-pattern. So in software, the thing that you don't want to do is the scheduler requires knowledge of everything that's in the lab. And we ended up having tables of data saying, here are all the experiments that I ran. And what we noticed is that over time, these tables got wider and wider and wider. As we said, well, wait a second, it seems to matter whether or not, what passage the pre-culture was on when we started. So we got to write that down too. And then we have QC, like did this pass QC? And then we want to know why it failed QC. And you get more and more and more columns. This is a known anti-pattern in data science, that if your tables are getting wider, something is not quite right. This should fail the sniff test. So we needed a better way to actually write down these data, and the answer came to us with chess. I don't know a lot about chess, but we have someone in the lab that plays chess. And what's interesting, going all the way back to 1949, Claude Shannon, the founder of information theory, had wrote a paper saying, well, here's how you could teach an AI to play chess. When you and I think about planning a chess agent, you might say, figuring out the state of a chessboard is pretty easy. You have a fixed number of pieces and a fixed number of board, and you just say for every space, what piece is on there? Is it a white pawn? Is it a black rook? And you can map out the state. Turns out that doesn't work really well because there's all sorts of rules in chess that I didn't know about that doesn't work. So you need to know where all the pieces are. You need to know which one has the next move. You need to know whether the king and rooks have moved because that determines if you can castle them. You need to have actually a back series of moves because if you repeat the same move within a certain amount of time, it's an automatic draw. You need to know the number of moves that have made since a pawn has been moved because if you make too many moves without moving a pawn, the game is also a draw. The state for chess is actually huge. There's all these other things that you need to go in order to figure out what are the legal moves in the next round. So chess doesn't actually record the state of their game. Chess has a really elegant system where instead of recording what's where, they just record all the moves that people make. So a chess game is fully determined by saying this is the starting state and here are all the moves. This is the white's move, black's move, white's move. And then they put all that in a chess database. So any game you look at, you'll get a series using this algebraic chess notation. That gets stored, and then they just replay the game. So if you want to know what did the board look like at move 12, they just replay the game, and then they can calculate what rules are allowed. And so they have a digital twin, in essence, a thing that just plays out the actual moves. So what Ben realized is that we should do the same thing in the lab. So we actually don't record the state of the lab. We don't record what's where or what's in what well. We just record every operation that happens in there. We record every liquid transfer, every plate movement, every time you change the temperature on an incubator. And nowhere in our database does it say, well, A1 contains this solution. It says, well, there was a transfer into well A1 from this other well. What's in that well? Well, I don't know either, but there were transfers into there. All we do is record the updates, just like in chess. And then we can reconstruct. So if you look at right now, our database is filled with millions or billions of every operation that's ever happened in the lab. And then we can ask, well, what is in well A1 at this particular time? And it goes through and it replays everything that happened. Said, well, you transferred out of this tube, and that tube was made up of this, and it reconstructs the state and says, I can tell you exactly what was in that well. And I can tell you what was in that well at every moment of time, and what temperature it was, and what it was exposed to, and how long it sat on the bench. If you record the low-level operations, you can get back complete state without storing any of the state whatsoever. Right? It turns out to be much simpler. And in fact, we solved another problem where it says, let's say I want to use an experiment and I have LB media and I'm going to fill a bunch of plates tomorrow. And I run it through the scheduler and it comes up with the plan. You said, that's a great plan. I have enough LB. That's wonderful. And I'm going to do that experiment on Monday. And then someone else comes in the lab and says, I want to do an experiment and I need LB. And the scheduler says, sure, you have 2 liters of LB right now. Right? And they say, that's great, I'm gonna do that on Tuesday. The problem is that person shows up on Tuesday and I used up all the LB on Monday, because we both planned looking at the bottle and says, well, there's 2 liters on the shelf, but they didn't know I was gonna use a bunch of it on Monday and they were gonna do the experiment on Tuesday. Well, it turns out that now is just a point of view. So whenever someone schedules something in the lab, we actually encumber all of those operations into the database. They haven't happened yet. When you plan, we add them into the database. And then when I go in and plan on Tuesday, I say, I'm going to run experiments on Tuesday, but plan it by reconstructing the lab out here in the future. It's the same procedure no matter where you reconstruct it. It's say, at the end of Monday, what's the lab going to look like? And it will know, it says, well, on Monday, all these things are going to happen. Right? So that LB is gone. You need to make more LB. It's on the shelf right now, but it's going to be used on Monday. And as things actually happen, we delete the encumbrances and we actually put the real thing that happened, just like your accounting system works. And I know that's not a ringing endorsement for the system. It's as simple as the accounting system. But when you file a charge, you say that money is reserved, it's encumbered. And then when it actually comes through, the amount is adjusted. Right? So you can reconstruct in the future. You can reconstruct in the past. You can say at any point of time what was in what well when you're doing the QC, just if you only record the operations and not the state. The last point on this level 1 planning is what we think when we're scheduling things out. This is a view of the top of a robot deck, and you say, well, we need to move between all these different wells and these different tubes. Like that's usually what we think about when we're doing our planning. In reality, and this point was already brought up, this is what we're scheduling over. So this is views in the lab. This is our freezer. This is inside the fridge. This is the wall of chemicals. We're scheduling from this. This into the robot is the hard part. And we had to extend the tracking system out into all of these. Now, we don't have automated solutions for this. The robots will say you need more LB. There's one robot in the system that's allowed to use called human, a very flexible robot. As a last resort, it'll say a human needs to make LB. Here's what you do, mix this many grams, this many water. But the human robot solves everything and it gets thrown in. We do not have cost baked into the use of the robots, but that would be interesting. The human and the robots unionized together. That would be quite the deal. But this is the hard part, like Henry brought up. Getting it from the freezer in is a bottleneck. Bottles are a bottleneck. That's why they're called bottles. I think that's right. Okay. We've actually found that depending on what you want to automate on the data processing side, sometimes you have to change the assay. So this is hot off the press. We grow bacteria in biofilms, and we've actually started growing the biofilms not on surfaces, not on plates, but we grow them on these beads. So these are hydroxyapatite beads. They're made of the same thing that your teeth are made of, because we study bacteria from the mouth. And you can see this is a bead that doesn't have a biofilm, and then you can see the biofilm that grows around the outside of the bead. The bacteria are fluorescent labeled, so you can see the microcolonies of bacteria that grow around here. Now, why would you grow them on beads instead of you can buy hydroxyapatite discs? People study these types of biofilms all the time. The only reason we put them on the beads is that it's easier to do in high throughput. So we can move the beads around more than we can move the discs around. When we're doing automated image analysis, the software is very good at picking out the beads. So we can find the beads, we can find the boundaries of the beads in the bright field, and then we can measure how deep into the biofilm we're imaging. Otherwise, we'd have to do some sort of autofocus and find where the plate is or find what the disc is. Right? So when people are saying we'd like to automate this assay, sometimes you need to reinvent the assay in order to make it more automatable. And that requires a conversation between the scientists who know what they want to do and the automation engineers that said, well, we could move a disc in and out, but that would be a real pain. Sometimes there's a different solution. Okay. Going back to this paper from this group in France, one of the striking things is they asked people before they started doing automation, what do you spend the most time on? And this is sort of a measure of how many people reported their most time-intensive tasks. You see doing laboratory work, which is the actual physical pipetting, the things solved by level 0 autonomy, right? That came in number 3rd. Most people spend their time designing experiments and then collecting and analyzing their data. So getting the robots in the lab didn't even attack the thing that required the most time from the scientists. This failed sort of the Pareto tests where you attack the thing that requires the most time. It's the experimental design is the issue. And so only when you get to level 2 autonomy, where you can have an AI system designing new experiments, do you really address the biggest time sink for most scientists, right? And unless you're doing a project where there's tons and tons, days and days of colony picking and pipetting, it's not going to go faster unless you get to level 2 autonomy, because the lower levels aren't going to help. Okay, so when you do this level 2 autonomy, to get it to run into a closed loop, you need 4 things to really get that to happen. One is it takes in a problem definition. So someone has to say, you're going to study this system, you're going to study these factors, these are continuous, these are discrete factors, you have to do that. And it has to say, this is the response, the thing that you're trying to optimize. And we can measure it. If you can measure it, then you can learn it. Or if you measure it, you can optimize it. So that's one. The second thing is you need a model, right? And the key is that all the model does is predict the response given the factors. So I'm varying these things in the media, and my response is the production of this bioproduct. That's what the model does. Predict the bioproduct in response to a particular setting of the factors. You need to decide, are you trying to maximize it? Are you trying to minimize it? Hector's team is fantastic at doing this type of thing for optimizing titers of bioproduct. We spend more of our time on characterizing, like doing basic science with the robots. You can do both. You just have to decide which one that you want to do. And then finally, you need some sort of mathematical technique to pick the next experiment. So you need a new model that says, given the data I've collected, what's the next experiment to run? This is all you need. If you have these 4 things, then you can run at level 2 autonomy into a closed loop. So I'll show you an example where we did this. So we said we're going to study the 20 amino acids, the proteogenic amino acids, and see what the oxytrophies are for those 20. So that those are the ranges. We're going to treat them as a 2-level range, so it's either gone or it's at 10 millimolar usually. And then we're going to measure growth. So we're going to pick the final OD at 36 hours. This is going to be the quantitative response for that. So that's part 1. For the model, we picked the simplest model you could think of, which is nowadays just a straightforward neural network. So given the 20 inputs, the amino acid concentrations, tell me what the growth is. That's all the model is. It's a black box. Right? You can use Gaussian processes. You can use any model that you want just to predict the response from these factors. Ours was to characterize, so we just want to learn the oxytrophies. We're not trying, but we could find minimal media, we could find maximal media. That's not really that interesting usually, but we could. And then we have a policy. We use this heuristic policy that says choose experiments to find small media that appear to be on the growth front, the border between it grows and it doesn't grow, and also an important constraint, we don't let it repeat experiments. Okay, so this is how we set up the problem. After you have the problem, you can just turn the crank. And in fact, the AI can just turn the crank if you break it down into these 4 issues. Okay, so once you get into this, I'm going to redraw now this closed loop that I've shown here. into actually all the steps that happen when you do this closed loop process. So we've already said that we're going to do, based on some manual growth experiments, we decided that the 20 amino acids were interesting to us. That decision was not automated. Humans made that decision. That'd be level 3 autonomy if we had a language model tell us, oh, you should check out amino acids in streptococci. That's interesting, right? To get started, we normally have a model that says, given our current data, here's the next experiment to do. At the start, we have 0 current data, so we have to do something. That's the techniques you use to pick the first round of experiments are usually different than you use for later ones. So we just chose random initialization, which works pretty well. There's nicer ways of doing random than really random, but it's a different set of techniques. Okay, so here is what our random experiments look like. So we did batches of 336 experiments per day. I'm showing you how many amino acids it chose to take out in search of this elusive growth front, and then the predictions that came back. So it's a solid dot if it grew, it's an open circle if it didn't grow, and then it's red if it got it correct and black if it didn't get it correct. So these are just randomly selected experiments. It's starting with the full media and moving down and randomly choosing them until it predicts randomly that it's not going to grow, but it doesn't really have any data. It gets about half of the answers incorrect. Okay. After you do that, then we actually run the experiment. So those are the predictions. We said 336 runs per batch. That's entirely based on logistical issues. We put 48 control experiments per plate. These are running 384 well plates. We put 24 positive and 24 negative controls, such that we have a positive and a negative control in every column, in every row, so we can correct the ODs, because we can't have a human look at it and say, I think there's edge effects or something. We need to have the controls are randomly placed, right? So that's done in this sort of verifying loop before the data are released. And then those data come back in and they're set to this very simple neural network. Again, that's all it's trying to do is predict the growth in response to the amino acids, right? So after we have the model, then we can plan what's going to be the next run. So we had to start with random when we had no information, and now we can actually plan a little better. So we use this hybrid policy where we start with all 20 amino acids, and there's one combination where there's 0 amino acids. We know it's dead here. We know it grows with the 20. We're looking for this sort of mapping, this line where you go from it grows to you took out an amino acid and now it no longer grows. Right? One way we do is we just randomly walk. We have the model go through. We keep taking out amino acids one at a time and checking with the model. Does it still grow? Do you think it still grows? And if it does, we take one more step. It still grow, and we keep on going until it hits this growth front and says, now I'm in a spot where it doesn't grow. So take that experiment and take the one before it where it grows and queue those up. So just a random selection. This is the exploration part of learning. Then we do some exploiting, where we say start at the top, and I want you to search for the smallest media that you can that still are on the growth front. So still a pair between predicted to grow and predicted to not grow. And the hard part for this is that it can't repeat experiments. So it has to keep trying and keep looking. And if it comes up with an experiment it's already done, it has to start over again. And then we increase sort of the randomness of this rollout policy to dig down. Right? So that's our plan. We explore and we exploit to pick the next set of runs. We pick another batch of 336, and now we're back in this loop. We can run those. We can retrain the model. We then search the model again. The growth front is moved because now it has better data. It's a little more sure. And then this cycle runs and runs and runs until we decide to call it off, until your budget is exhausted. Okay? That's not a joke. Like, you have an experimental budget and you run until it's exhausted because for convergence. Local budget, not like EMSL's budget's gone, and then this is going to be one experiment. So let's watch it go. This was day 1. This was the random experiments. I can show you every other day. By day 3, you see it's already a lot better. You can see this are sort of the random searches. This sort of cluster is the deeper dive ones, and a lot less red circles. Now this is day 5. The accuracy actually went down. This happens. It thought it knew what was going on and got some data that contradicted it, and now it says, oh, I didn't really understand the system as well as I thought, and it moves back. But around by day 9, day 10, it's over 90% accurate. So the model can pretty much tell you ahead of time if it's going to grow or not. And at day 13, we just stop it. Out of budget. We're done. So now we can just ask the model, is it going to grow in this media or not? The model has learned it entirely. We could have done the same thing where we had an objective to maximize or to minimize. Here, the objective is just learn the entire space as best as you can, but you can do process optimization in this way as well, the same loop. Okay. Now, the last step here is that after we've run this loop and now we're happy, we have this really great model that can predict. The model is a black box, and now we can interpret. So we run this as a separate process where we take and pull out sort of a logical rule. So we have a second machine learning system that says, build me a simple rule that a human can understand. It says, for one of these bacteria, here is the amino acid oxytrophy pattern. It's this rule. The rule is actually pretty accurate. If you wanted the real answer, ask the model, but this is something that the humans can understand. Interestingly, we had a grad student look at a smaller set of data that they did by hand. They came up with this rule. So the rules were a little different between the two. So anyone want to vote? Who says the AI was right? Who says the grad student? It wasn't Ben. Don't feel bad. It's not a different grad student. Who says the AI? Who says the grad student was right? So if you look at the manual data from the grad student and compare both models, this is wrong. This oxytrophy doesn't show up. If you look at all of the experiments collected by the system, then the AI is right. So it's not that the grad student was incorrect or synthesized the data improperly, they just didn't have enough data. This oxytrophy only shows up after you take out a number of other amino acids. It's in small media. So the gold standard data we have does not contain all of the oxytrophies, and that's why the grad student couldn't see it. So collecting more data, even just in an exploration type fashion, can move the gold standard and move your interpretation of the system. So when in doubt, if you don't have anything else to do with AMP2, just random experiments, just explore. You never know what you're going to find out. Until you run out of budget. That's the okay. The cautionary tale on this. Do not try and get too fancy. This is the one thing we have learned time and time again, is do not try and play all sorts of tricks with the cycle. The simple stuff works, and it works really well, surprisingly well. Originally, we had this grand idea. The formatting on this might be a little messed up, so bear with me. The idea is better than the formatting. So we thought you could think there's one media that has all the amino acids, and then if you take one out, you sort of branch it into this tree, where there are 20 ways to take one amino acid out. And then in each one of these leaves, you can expand it by taking out a second amino acid, and then you get 20 more amino acids that comes out from there. And it goes on and on again. There will eventually be 2 to the n leaves, terminal leaves in this tree. That's over 1 million possible combinations of the amino acids. And so then we had this brilliant idea. It said, if you find anywhere in this tree a spot where it doesn't grow, you've taken out amino acids and it's dead. Taking out more amino acids is not going to bring it back to life. So you can stop exploring the subtree that goes below it. So everywhere we get no growth, we can just prune the tree, and then we can really reduce the search space. This is called branch and bound in the combinatorial optimization space. Right, so we can do this, and now the problem becomes much more tractable because we have this really clever thing. We call it metabolic convexity. If it doesn't grow in these 10 media, there could be another media with 9 different ingredients that it grows in, but taking away something from that 10, a strict subset, isn't going to bring it back to life. So this was really clever. It's not right. It's wrong, actually. So biology does it. So here's an example. So when I'm showing here the fitness, the larger the circle, the more it grows. So when you take out methionine from this bacteria, from the meat of this bacteria, they don't grow. They don't like it very well if you take out methionine. You thought, oh, so I've taken out one amino acid, taking out something else is not going to bring it back to life, and it does. So if you also take out cysteine as a second one, it grows much better. So depleting both of the sulfur-containing ones, presumably a regulatory thing. It senses the presence of either and shuts down sulfur amino acid biogenesis. So, but it still needs the other one. If you take out both, now it kicks on its own systems as well. So metabolism not convexed. We found this on day 1 when we were trying the new policy, that we were trying to be way too clever. Just let the robots win. This is not rare. It looks rare because none of these circles are showing up. So this triangle in my head has lots of circles, and about half of them are blue. And the blue circles are non-convexities, things where it grows better when you take away. It's beautiful. Trust me on this. I'll have printouts in the lobby afterwards, and you have to be convinced of this. This is all the data we've collected with the robots. Is these 7 points right here. Anyways, the other thing, you shouldn't get fancy on the modeling side, right? So one thing we've learned from all these big breakthroughs that have happened, beating a world champion in Go, folding proteins, we can have it talk. Like the one thing all of these have in common is that they took a black box model that really didn't have much domain expertise. They threw a ton of data at it and they got useful predictions, right? That works really well. In fact, that's really the only AI triumph we've had for emergent human type level intelligence is black box model plus massive data, not getting over fancy on the models. With one exception, one key exception. There is an important role for having mechanistic models. So here's the sort of black boxes we have. We put in nutrients and our neural network or our Gaussian process outputs the growth and we don't know how it does it. And that works really well for collecting data, for just driving the robots. It would take you too long to build another model to drive the robots. You could have done a million experiments in that time to begin with. What this is not good for is to plan experiments that use multiple different modalities, right? So when I have this model, I can ask, what is the most informative growth assay to do? Tell me the nutrient combination for it, and I can get that. There are good policies that let me do that. If instead PNNL offers to say, well, what if we want to do proteomics? What would be good proteomics? I would say, well, from my black box model, I have no idea because it doesn't have proteomics as an input. This model, all of my data can't tell me anything about the proteome and the uncertainty in the proteome, right? Until I start doing proteomics experiments. I'd be doing random proteomics experiments to begin with. So if you're doing multi-omic type thing, then having some sort of model, this is a genome scale metabolic model right here that takes genetic information, enzyme information, and nutrients. Even if we're only manipulating these and measuring these, the mechanistic model can be that link in between the two. So metabolic engineering, things where, oh, I can make knockouts and I can change the media, that's the role for mechanistic models. If you're just straight phenotypic profiling, you don't need the genome scale model to start. You can bring that in after. We've had a lot of success actually combining the two. So we have genome scale metabolic models that have neural networks that fix the models. So we measure growth data, we manipulate the growth environment, we measure growth, and then we use this to train a neural network here that learns the regulatory interactions and that actually improves the metabolic as well. So we can kind of sort of mix the best of both worlds. We don't have to build mechanism into here. We can just learn that and we still are able to do the multi-omic thing. The mechanistic models can also save with experimentation costs, so the experiments are really expensive. That'd be the other case where it comes in. But other than that, the black box models are easier to get up and running. Okay. Remember that when you're doing these experiments, this is the formula. This is really the only math in this talk. So the throughput of your system, you have to multiply that. How often is the system running? And then the information density, how good was the experiment that you designed? That's the amount that you're going to get out of your system. I toured a pharmaceutical company one time and they had a video monitor showing the robot running because it was never up and running because they had to analyze the data from the previous campaign before they ran it again. Like if you can do 60,000 experiments a day and you run it once a year, that's 60,000 experiments a year is your throughput, not 60,000 a day. And if you're doing things that have low information density, that actually doesn't help. So you need to focus on all three of these when you're getting good data to come out of your system. The last sort of level of autonomy was level 3. So we don't do a lot of work on level 3. I think it's very exciting to say, can a language model tell you what's important to do? But the only thing I would caution on looking at this last level of autonomy is that I think it's great, and I think language models will be really useful for this, but do not have machine learning systems that cross the levels of automation, right? You can use language models at separate stages of this, but they should be separate models because we have had systems, our level 2 system that also emitted instructions for the robots, so mixing level 2 and level 1 autonomy, and we have always had to rewrite all of the code to re-separate things. The robots stay separate, the scheduling software is separate, and the level 2 autonomy, the modeling is separate again, and they have defined interfaces between them. But as soon as you entangle them, they are not modular, they are not reusable, and we spend a year pulling it back together and ripping it apart. So Ben in our lab is the only person that works on level 1 autonomy. all of our students that do level 2 autonomy output experimental designs that go into Ben's software. They don't touch anything. They don't know how the robots work. This is very confusing during qualifying exams to say, I don't know, we output experiments. They go into software written by Ben and data comes back. And there's robots somewhere and I think they're running. That's going to be, your P&L is going to make so many engineering students qualifying exams difficult because experiments just happen in a black box. Thank you for that, Scott. So keep these things separated, I beg you. If you do nothing else, keep the separation. One thing this allows you to do is we used to do science this way. Humans design experiments and then they run the experiments. This is sort of what we're talking about now, this automated science. AI plans the experiments and then robots run the experiments. Keeping the separation allows you to do the other two combinations. So we can reuse robots in the lab even when humans plan experiments because they're separated. Oh, here's a design. It doesn't matter if a human gave it to you or an AI gave it to you. We know how to get an experiment onto the robot to feed it into the level 1 system. And this is sort of the big game. I think there's a huge opportunity for having AIs that plan systems and then humans carry them out because it's much faster to spin up humans to do experiments, especially very difficult, very expensive experiments. There's a lot of, fantastic capabilities here at EMSL that require humans to do that can't be automated in the near future, but can we still get AI to suggest these are the conditions used in profile, even if that gets sent across the hall and humans are involved and carry them out? This is where the big wins, I think, will be in the short term in AI-driven science, not as much this. Okay, so in conclusion, my summary, like the key points for every level of automation here. So as soon as you bring robots in the lab, you've got to be prepared that you're going to spend your life watching them pipette and watching them move plates it's like being with a little kid. It's like they got a spoon in their mouth and only spilled half of it on their face. Like, good job. Like that, you're going to think, you got it in the plate reader. And it was amazing. It was only partially kiltered. Like, that was great. When you're at level 1, the lower level operations, recording all of those will avoid all sorts of issues with recording state. And I think that might be my most controversial statement, especially mapping to a limb system that is only about state. When you get to the level 2, the simpler you can make it, the better. Simple black box models are a great place to start. If you don't have the mechanistic model, the mechanistic models have huge advantages once you have them, and no fancy policies. Don't try and overthink the system, just let it run, especially when you have scale like you do with AMP2. And finally, level 3, I think, is coming, and we should be working on that, but all of the levels have to keep isolated or you're going to regret it later. I want to thank you for the wonderful invitation to talk here. Thank all the people who helped do this work at Michigan, and a lot of the work I presented is Ben's, and he's here to talk. And of course, this wonderful partnership, I always love coming out to Richland and talking to the PNNL people. So thanks so much.
Thank you, Paul. Are there any questions? Oh.
Paul, I have oh, sorry.
There's a line. Just
So I have a question for you about the level 1 and maybe replicating level 1 across facilities. So if you're recording the operations and not the states, and that's, if we use the chessboard analogy, the reason that chess can just record the operation from place to place is the chessboard stays constant. Is there a model to maybe take the initial conditions that are in a lab and take the operations and apply them to different initial conditions? Sorry if I'm not being clear on that, but I'm thinking about how if you record the operations but not the states, you're able to then have learnings that transfer across different facilities.
Yeah, the sort of two notes to that. One is there is like a very initial state. So there's a special thing whenever we bring a reagent in from Sigma or when we make a plate, when it's sort of burst into existence, it's originally signed a location, and then everything gets moved after that. So there is like a Big Bang-esque thing for everything that's in the lab that assigns it. The other thing is that the operations are really robot agnostic. So the operations, for instance, are transfer, like you move liquid from one to the other. That transfer looks very different on our robots, whether it's done with a tip transfer or a non-contact dispenser out of the tube. That is all handled on a separate side where Ben's scheduler emits protocol-specific things to the different robots. So I think the protocols can be transferred because if you move to a different lab or we switch out one of the robots, you can actually recompile it to emit instructions for a different machine. Say, I want to make these same transfers, but the Tempest is down. We're going to do it on the Hamilton instead. And I think that's nice because you know then that what you're getting is the same final state. I get the same concentrations in the wells, even though some have a dead volume, so you lose some liquid. The amount that actually is transferred might be different. You're going to make sure you end up in the same thing, which means if you're trying to do reproducibility, you should get the same result. So we're hoping that works for us, that we're independent of the robots, but I think you're right, it could be applied lab to lab.
Quick question here. Thanks, Paul. This is great. Always very crisp. One question I had, and we think about this a lot, is information density. Always you want to cram the most you can from an experiment. This seems a little counterintuitive to no fancy policies and just let the robot run. Could you maybe talk a little bit more about where that threshold is for you?
I don't think those are orthogonal. I think the no fancy policies, if you use an off-the-shelf Bayesian optimization policy, it's going to be generally high information density. It's going to say change everything every run. And we end up usually having to constrain it because, for instance, when we make genetic manipulations, we don't have the same freedom. We can't delete half of the genes in every organism. We can maybe delete 3 or 4 with our technology. So we have to put some logistical constraint on the policy. But a simple policy is not the let's try one amino acid at a time. It's simple policy in the Bayesian optimal sense. So off the shelf might have been a better way to phrase that. I agree.
Yeah, in terms of planning or record the operations, not the state, that, when you're doing chess, when you move, up and down, it always happens and it's quantized. There is no error there. But in biology, there are many things that are, probabilistic, right? I put my CRISPR MAGE, with my plasmids and in my electroporation, I may get a mutant or not, and I need to assess the state, right? Just knowing that I've done this, and that doesn't really ensure that I understand it and what I get at the very end, right? So in those cases, with more fuzzy places where there's more errors and there is an stochastic result, we need to assess the states. That you can't assess every state, but what you can do is like, we have this level of evidence for this at this time, at that time, right? Otherwise, the operations is just not enough to plan what's going to be the next step because you don't know what your state is, right?
Absolutely. So we push that uncertainty onto the modeler that wants the data. We say at any point you can say exactly what happened inside the lab, but what the implications of that, like you're saying, that we still leave up to the human. One thing we also separate is that when biology gets involved, we handle that as a special case. So when you can say, I know exactly what's in a media up until this point. And as soon as you put a biological ingredient in there, like a microorganism, the scheduler gets a little iffy about saying, I know exactly what's in this media again. Because if it's growing, it's presumably eaten something and produced other things. And it handles differently. Like if you transfer any small amount of something that had E. coli in it, now E. coli moves to the other one. How much, we're not really sure, but so we do that where right now we say at some point when a reaction happens, either a chemical reaction or biological, we have to flag it and say there's uncertainty here, and it's left to the human to sort out and say, yes, this could be different. You're exactly right.
I guess my point is that in the real world, beyond just like doing media, uncertainty dominates.
Yes.
Hi, 2 questions actually. The first one building on the what Hector just said. How do you deal with if something went wrong? Like when you mentioned, like you might not actually aspirate in the part of the automation. So say your record of operations says, well, A1 is supposed to have 10 microliters, but in reality, you go and you look and it doesn't, and you don't know where it went wrong. How do you deal with that?
So when we plan things out and say, here are the future operations that are going to happen, those actually don't become things that happened in the past. So those get removed, and then the real thing gets uploaded as this what actually went down. So if the technician observes something happens, they can say, no, put everything in except for this operation that never happened. And then when we reconstruct, we account for that. The other thing is if we go back and say, wait a second, we bought the wrong thing way back. When we make a change in the database, we apply it at a particular time point. So you can actually reconstruct based on what we thought it was at a particular standpoint. I can say when we reconstructed on July 4th of last year, here's the answer we got. But now that we've applied this change to say it was sucrose, not glucose, it looks different now. So it kind of like runs like a Git repository where you can change anything, but it doesn't remove the previous state. It just updates for the future.
Right. But like, say if you don't know what went wrong, somewhere along the line, something got missed.
Right. We have to rely on the QC to sort of flag and say this nozzle went bad, so we don't know, and we can take the data out. And then the only other for high throughput where we're not looking at everything, all we can say is that in the future, if we had some way to discover that something went wrong, we could incorporate that information. But we can't guarantee that everything we say is going to happen in the lab happened in the lab because of the robots. Yeah. You're right.
And a totally different question about the AI planner scheduler. Did you I think the other thing that's hard for AI to do is actually get meaning out of the results, right? Like you have, oh, these amino acids are necessary in this combination for the bacteria to go. Have you found anything like really surprising from the data that you wouldn't have expected based on just like if a human was thinking, oh, it'll probably need this?
Yeah, what we found is that our genome scale models are really bad. So we built models of what got us started on this. We built models of 4 streptococci, all live in the same niche, and that none of them have any predicted oxytropies. They have complete biosynthetic pathways for all 20 amino acids, and the models are manually curated and checked. Each one of those species has a completely different pattern of oxytropies. And they only live in your mouth. And the robot was able to find all those and map them out and say, these are the rules for all of them. It doesn't understand either, but it can find it. But we were stumped, and we have 4 models. They're all different, and there's no way we could look in the genome because we didn't have any regulatory information. So those are the types of things where had we not done all the experiments, we wouldn't have been able to see that.
We have an online question from Ratna.
Thank you. Thanks, Paul, for the
speaker in here.
Can you hear me?
Yeah.
Yes.
Okay, thanks. Thanks, Paul, for the exciting presentation. We are looking forward to this, opportunity. Taking your chessboard analogy, so there are only 32 pieces as an input. When you compare it with science experiment, the reagents are more than that. So, which means it results into a multiple combination states and operations, right? How do you contain this boundary?
Right. So the reconstruction actually turns out to be pretty efficient. So every compound, every chemical, every well, every piece of plastic in the lab is in the system and recorded at some point. And we thought it was going to be difficult to say, all right, if I want to reconstruct this one well, I have to map all the way back to the start of time. It turns out to be really fast, and then also Ben has a caching mechanism. So the database saves state temporarily through, and it goes back in the reconstruction until it finds a cache and then uses that to come forward. That doesn't change the theory of how it works, but it makes things a lot faster. And then it also knows when it has to update the caches when you edit things. So Ben can show you all these drawings about alternative timelines and how it resolves this multiverse, but computationally we haven't hit a bottleneck yet, even though, like you said, it's much more interesting to play biology than to play chess.
Words of wisdom. More questions? Questions for Ben? No, I'm just kidding. Oh,
So you said that the genome scale models failed in finding these isotropies. I guess, have you thought of ways to leverage the best of mechanistic models and AI to really modify these models in a systematic way by using the new data so as to be able to predict what you find experimentally?
Yeah, it works really well. So this type of framework here, where we take the genome scale model and we just make a very simple neural net that can, in response to an environment, change the upper and lower bounds of the enzyme activities. As soon as you apply that and show it the data, it can now predict everything. Just by changing in fact, just the upper bounds, because it's enzyme activity. It's not directly reaction flux. We pull the genes out. So because it's a regulatory thing and it's an interpolation for the model, as soon as you get enough data, 5,000 or 6,000 combinations, now the genome scale model is correct. So yeah, you're absolutely right. That works really well.